Okay, guys, this is week 2, part 6. We're going to talk about the tubes, okay? So we have all these different color tubes. Each one has special additives that do specific chemical things to our blood, okay? What is an additive? An additive is any substance added to the blood collection tube. That's important to know, okay? An additive is any substance added to the blood collection tube. Are all additives the same? Absolutely not. Some keep the blood from clotting, which is an anticoagulant. Some help the blood to clot faster, which is a clot activator. Some additives separate the cells from the liquid portion of the blood. Okay? What is a characteristic that all the tubes have in common? It is that they have a pre-measured vacuum that pulls the blood into the tube. Okay? These tubes have stoppers on the top that correspond to the additive it contains. That means the color of the stopper on top of that tube is going to tell you what's inside of that tube. Okay? Let's talk about serum versus plasma. So, serum is the liquid portion of clotted blood after we centrifuge. What is centrifuge? That's when we put it in the machine and it spins at like 30,000 RPMs for a certain amount of time. And it separates the cells or formed elements from the liquid portion of the blood. So blood is naturally going to clot in about 30 to 60 minutes. When we add a clot activator to the tube, though, it clots in 5 to 10 minutes or 10 to 15 minutes. Normal fasting serum is pale, clear yellow fluid. Okay? It has the same components as plasma, except it does not contain fibrinogen. That got cut off there, so I'll write it in. Okay? Fibrinogen. Does not contain fibrinogen because fibrinogen is a clotting factor and it was used when it formed the clot. Okay? Plasma is the liquid portion of anticoagulated blood, or blood that has not clotted. It is whole blood. It's the same form as it's flowing through your veins. Whole blood is. Plasma is also pale, clear yellow fluid. That's why we say that you cannot visually tell serum and plasma apart. You cannot look at it and tell which one is which. Okay, you have to know what tube it was drawn in to be able to tell if you have plasma or serum. Plasma does contain fibrinogen and the other clotting factors because they were not used in the clotting process. Not all tests can be performed on serum. Most can, but like our coagulation tests, they can't be performed on serum because the clotting factors are used in the clot, right? So we can't measure them. Some chemistry tests like ammonia and potassium cannot be performed on serum because clotting releases the substance into the serum and would give us falsely elevated results. Now, this is for your FYI, okay? Some tests are needed stat, so we can't wait 30 to 60 minutes for the blood to clot, or even the 15 to 20 minutes. If the blood is prevented from clotting, many factors are preserved, and the specimen can be centrifuged immediately and tested right away. Serum is the specimen of choice for immunology and serology because of the chemical makeup and accuracy for these tests. So all this is telling you is giving you a little bit of the reasons why we need both plasma and serum. Okay? Some tests can't be performed on plasma or serum, and we need whole blood. Adding an anticoagulant to the tube creates that whole blood specimen. So we kind of looked when we were in lab the other day about the 3 distinct layers. You do need to know these. When a whole blood specimen is put in the centrifuge or sits in the rack and gravity takes hold, you're going to have 3 distinct layers. You're going to have plasma as your top layer, white blood cells, platelets as your middle layer, and we call that the buffy coat, and then red blood cells are going to be your bottom layer, okay? When we go to test this, though, we're typically going to be mixing these for 2 minutes if we're doing a whole blood specimen, okay? Some tests require whole blood, like a CBC, because we want to see what it looks like. Those red blood cells, white blood cells, and platelets, we need to see how many you have, what may be going on, and we need it to be flowing as if it's flowing through your veins so we can get an accurate measurement. Because if we just, if we didn't allow it to flow, it would just kind of sit there and we would get an abnormally large number of cells, right? An additive, again, is any substance added to the blood collection tube. The type of additive required for blood collection generally depends on the test being ordered. You cannot substitute or combine tubes with different additives. No mixing of blood specimens, even if the additives are the same, because the excess of additives can be created and negatively affect the test. So if you get a tiny bit in one tube and tiny bit in another tube of the same color, trying to troubleshoot the venipuncture, just do the best you can, you would just send both of those tubes to the lab, okay? Because and I would put like 1 of 2 and then 2 of 2, just so that the lab knows, like, it's a hard stick. You could even call down there. You could walk it down there yourself. There's all kind of ways to let the lab know it was a hard stick, and you're sending both tubes. So they would sample out of each tube and they would not combine the two, okay? Anticoagulants, clot activators, thixotropic gels, that is that barrier, that jelly stuff at the bottom of the tube, that creates the barrier between the cells and the serum or plasma whenever we centrifuge the specimen. We call these a serum separator tube or plasma separator tube if they have that thixotropic gel, okay? Additives come in liquid, spray dried, and powder forms. We should always invert our tubes immediately, no matter what the tube we're drawing. This is just going to make sure that everything is well mixed, okay? We never vigorously shake a tube because it can cause hemolysis. Hemolysis is a lysis or destruction of the red blood cells, okay?